Good morning, everybody. It's our great pleasure and honor to welcome Professor Harold Varmus at AUBMC. Professor Varmus is a great scientist whose achievements and discoveries in the medical field were remarkable enough to win a Nobel Prize in 1989. Dr. Varmus is going to tell us today what it took to reach there, giving a description of the scientific process he had followed, which led to magnificent discoveries. Currently a professor of medicine at Cornell Medical College, Dr. Varmus is a former director of the National Institute of Health, NCI, National Cancer Institute, and Memorial Sloan Kettering Cancer Center. Being an advocate for open access publishing and data sharing, he established the PubMed Search Center and co-founded the Open Access Publisher PLOS for Public Library of Science. Dr. Varmus was appointed by President Barack Obama as a co-chair of the President Council of Advisors on Science and Technology. In addition to the Nobel Prize, Professor Varmus has received many honors for his research. He has authored over 400 scientific papers and 5 books, including an introduction to the genetic basis of cancer for a general audience and a memoir, The Art and Politics of Science, published in 2009. Dr. Varmus' visit, sponsored by the Carol Bellamy Seminar Series, was initiated by our student Natalie Nasruddin and later supported by the Biology Department, the Faculty of Arts and Science Dean's Office, the Faculty of Medicine Dean's Office, and the AUB President's Office. Dr. Varmus, we are honored to have you with us. Please join me in welcoming Professor Harold Varmus.
Well, thank you very much. Hoping we get something going here. We're not connected. Okay, good. That's what I want to see. So, first of all, I want to thank you for that very concise and accurate introduction. I want to again thank Natalie, who has been arranging my extremely enjoyable trip to Beirut. This is my first visit to Lebanon, and I was not surprised to find how complicated this country is, but I'm appreciative of the chance to understand it better and to have seen remarkable historical sites in many of your cities. I've now been north and south and east, and I think I have gotten to know things quite well and have enjoyed meeting your faculty. Those of you who were present at my first lecture in this 2-lecture series last Friday know that I have a deep connection with this school because I was an undergraduate at Amherst College in Massachusetts, and Amherst College is the alma mater for both Presidents Bliss, who founded this school in 1866 and ruled it for 54 years. I did know personally quite well President Calvin Plimpton, who was here in those difficult years from 1984 to 1987 as your president and had previously been the chairman of the board of directors. Those of you who were here on Friday, not in this room, but in a different room on this campus, will recall that I spent time talking about several, indeed 9 axioms by which I have come to enjoy a life in science. And today, my intention is to tell you a little bit about the kind of science that I've done. I will spend a little bit of time talking about work that led to the discovery of cellular oncogenes, the work for which my colleague Mike Bishop and I received the Nobel Prize. But I will mainly be talking about work that's been done fairly recently, and my goal is to contrast the kind of work we did then and the work we're doing now, and to make some more general points about the nature of scientific work. So, what I will try to do is to explain the narrative behind the kinds of experiments I'll be talking about. I hope we'll have a chance for questions and answers afterwards. I will be showing a fairly limited amount of primary research data, mainly trying to explain to you why certain kinds of experiments were done, what kind of data could be obtained, how that data was interpreted, and how efforts to understand one thing lead to new questions about how science works and how cancer cells develop. So, as I explained on my lecture on Friday, in my view, and I think a view held fairly generally, science tends to begin with a good question, and people sitting in a classroom learning about the past are never as excited about science as are people who are presented with a question about the next step in discovery. But efforts to try to answer questions are inherently limited by the kind of technologies that are available for answering questions. And I tried to explain this on Friday by describing the way in which, in my early days of research at the NIH, I learned that having a new technique allowed me and my colleagues to begin to answer questions about very simple experimental systems that led ultimately to questions about more complex systems like human cancers. So one question has dominated my research career, and that's the question of how a normal cell becomes a cancer cell. And I just illustrate this with a couple of simple illustrations. They don't mean much except to say that have people been able to hear what I've said so far? Yes, I hope so. Thank you. Well, the fact that you're laughing suggests you've heard that. Shown at the top is a normal cell with a nucleus and cytoplasm behaving, we think, in an ordinary way. And below is shown an illustration from a magazine in which an article about developments in cancer appeared, showing a cell that is growing at a more rapid rate than other cells, able to invade its surroundings and do other things like recruit a vascular system to help provide its nutrients. So cancer cells have many properties that are not inherent in normal cells, and one of the challenges here, as indicated by the question marks, is to understand the changes that occur in a cell to convert it to a cell that grows inappropriately, forms a tumor, invades, spreads to different distant sites, and ultimately will kill its host. And I suspect that just about everybody in this audience either knows enough about cancer to fear it as a disease, has experienced the loss of friends and relatives who've had this disease, and understands the compelling argument for scientists to try to understand this conversion that results in so much loss of life, and that has been an underlying precept for me and many colleagues who have worked on this problem. But inherently, we are limited by the kinds of techniques that are available, the materials and methods that scientists use. And when I began doing cancer research, I hate to say it, nearly 50 years ago, we had some tools to work with, but they were fairly limited because we didn't yet have the recombinant DNA revolution. We didn't have the human genome to study in the kind of detail that's available now. There were some viruses that cause cancers in animals. As you'll see, that's important. We had the ability to grow cells that came from cancers in a petri dish in culture. We had a few human cancer cell lines available. It was possible to detect certain genes by using molecular probes to ascertain the presence of certain genes in cells. But it wasn't until considerably later, 1 or 2 or 3 decades later, that some of these new methods that are so important, such as recombining DNA to study it after growth in bacteria, the appearance of DNA sequencing methods and the ability to insert genes into mice to observe the behavior of genes, only then were we able to begin to understand the genetic origins of cancer, which we now know to be at its heart a genetic disease, one that is dependent both on the inheritance of certain genes from one's parents, but more importantly, as a result, changes in genes that result from exposure to either the natural processes of DNA replication and cell division or exposure to mutagenic events that occur in the environment. We now have a much more powerful array of tools. I'm not going to review all of these, but simply point out that there are more sophisticated ways to grow human cancer cells, either in culture or in mice. There are many more human cancer samples that have been subject to analysis, and there are new so-called high throughput methods available for studying the DNA, RNA, protein, metabolite production, other features of normal and cancerous human cells. So I like to think of cancer research as having occurred historically in three major phases. Of course, cancer was recognized as a disease thousands of years ago, but until the mid-1950s or so, it was largely an observational science with a few exceptions, one of which I will point out to you in a moment. Then there was a phase between the 50s and the 1990s when molecular biology grew in the wake of the discoveries by Watson and Crick and others of how information is encoded in DNA and how it's expressed through RNA and protein, and combined with knowledge about animal cancer viruses, it was possible to carry out experimental work on cancers that gave rise to some fundamental concepts about genes that are subject to the kinds of changes that lead to the development of cancers. Since roughly 2000, the period I will refer to as now, as opposed to then in this talk, we have begun to do data collection, do experiments at a much higher velocity, and with the generation of much more copious amounts of data requiring new methodologies like computational biology to try to understand the vast numbers of changes that contribute to the generation of a cancer. But I want to start this, the substance of this talk by going back over 100 years ago to a discovery that influenced my own life and has been fundamental to understanding the nature of cancer as a disease that arises through mutations in genes. And that event was the discovery of a virus we now call Rous sarcoma virus. In roughly 1909 or 1910, a chicken farmer on Long Island brought to a young physician at the Rockefeller Institute named Rous a chicken with a tumor called a sarcoma, a cancer of soft tissue that was occasionally observed in domestic chicken flocks. Rous did an experiment that led over 50 years later to the award of a Nobel Prize to him when he was in his 80s. And the basic experiment he did was to take that tumor, a sarcoma, grind it up, pass its contents, the extract through a filter that would retain any cells or bacteria, and he found that what emerged through the filter, the filtrate, when injected into a new chicken, produced a cancer. And he could repeat the experiment, taking the new cancer and doing this again. And although we knew very little about viruses at the time, this met the criteria of being a filterable agent that was able to cause disease when introduced into a naive animal. So the material in this filtrate, which came to be called a Rous sarcoma virus, proved to be a virus of the class we now recognize as retroviruses, and everybody in this room knows a lot about at least one retrovirus, HIV, but this is among the very first retroviruses to have been identified. Now, work with this virus was really quite limited for a long time. People repeated the kind of experiment that Rous had done, taking the Rous sarcoma virus and putting it into a variety of animals and seeing that tumors appeared. And this was useful at one level, but many people said, well, we know that human cancers are unlikely to be a result of infections. There are indeed exceptions to that, but most people said this is not really teaching us very much about cancer. A major advance that led the way to the kinds of experiments that many of my colleagues and I did in the 1970s was an experiment that allowed a simple test for the cancer-causing properties of Rous sarcoma virus, an assay for the carcinogenic effect of the virus. This work done by Howard Temin, who many of you may know about as the discoverer of the enzyme reverse transcriptase, and his colleague, Harry Rubin at Caltech, made a simple test. And that test was based on the use of a chicken embryo, the disassembly of the embryo and the plating of cells in a dish to form a monolayer of fibroblasts, then the addition of that filtrate that contains the virus shown there allowed the counting of particles that could infect a cell and turn that cell into a cancer cell, shown by the group of cells that have emerged from a single infected cell that have a different morphological appearance and have overgrown the layer of normal fibroblasts. So it was possible to say that any single tube of Rous sarcoma virus fluid had a certain number of infectious particles that could make a normal cell behave like a cancer cell. And that opened the way to a lot of important experiments, perhaps the most important of which was performed by Steve Martin at Berkeley, then a postdoctoral fellow, generating a temperature-sensitive mutant that could transform chicken cells only at a relatively low temperature, 35 degrees centigrade, and not the higher temperature, 41 degrees. This suggested, because the virus was still able to grow but could not change the behavior of cells, could not transform cells, that there must be some function in the Rous sarcoma virus genome, which we then knew to be about 10,000 nucleotides of RNA, and that that function was required to initiate the transformed state. Moreover, by carrying out the shifts in temperature shown on your right and observing that a transformed cell would become untransformed if you raise the temperature, and transformation would reoccur if the temperature were lowered. So this suggested that the function of the virus that was responsible for transformation was sufficient to transform and required to maintain the transformed state. And these are concepts that are important today, because if you think about the ability to try to treat a cancer, this kind of experiment teaches you that continued expression of a cancer gene, in this case a gene carried by a virus, is required to maintain the transformed state. And that's a fundamental principle that underlies one modern form of cancer therapy. Now, this kind of experiment, of course, raised the question of trying to identify the gene that the virus carries that's responsible for transformation, to ask whether the gene is related in any way to normal cellular genes in humans that might be important for generation of human cancer, and to ask what kind of protein might be encoded by that gene and might be a way to start thinking about human cancer more generally. So here I have to summarize a great deal of information that was generated during the 1970s, but let me just show you a few cartoons that illustrate what was learned from this kind of work. First of all, it became apparent that Rous sarcoma virus and many other retroviruses that were found in other species that were nevertheless able to cause cancers in mice and other rodents and chickens and other birds, had at least 3 genes that are required for the growth of the virus, and those genes, as in other viruses, are required to make the coat proteins and the enzymes that are found in a virus particle. But Rous sarcoma virus contained another element called the sarcoma gene, or SRC gene, that was the gene that experiments like the temperature-sensitive mutant experiment I just described showed was essential for initiating and maintaining the transformed or the cancer-causing phenotype. Mike Bishop and I and our colleagues, showed that if you made a molecular radioactive probe for that gene, that you could find a very similar gene in normal cells. And that gene, like other cellular genes, was formed of coding sequences interrupted by nonsense sequence, exons interrupted by introns, and the gene encoded a protein of about 60 kilodaltons. So that suggested that at some point we had learned something fairly important from the study of this chicken virus able to cause sarcomas in birds, that the virus carries a cancer gene, and that gene encodes a protein of a certain size, which we and others contributed to understanding by learning that that protein is actually an enzyme that carries out a very important function. It adds phosphate by transferring it from ATP to tyrosine residues in proteins. Secondly, we could see from experiments, or from the kind of concepts that emerged from experimental work that led to the previous cartoon, that the gene was derived, some might say stolen or pirated, from a normal cellular gene and was altered in subtle ways, in the case of Rous sarcoma virus, by the changes of a few nucleotides and a small deletion of certain nucleotides to make this enzyme behave differently from the normal enzyme. So instead of being a nicely regulated enzyme, it became constitutively or unalterably activated and was acting as a highly powered enzyme transferring phosphate to tyrosine and other proteins at an inappropriately high rate. And then we learned from work done by our group and many others that the other kinds of retroviruses that were found in birds and rodents, and even in some cases primates, also carried genes derived from normal cells, and those genes were again altered in interesting and significant ways and were able to transform cells. And here's a cartoon from the 1980s showing many of my colleagues. You may note that they were almost all males, disturbingly, but they were working with viruses from a variety of animals, all of which contain genes. I draw your attention to a couple of them, the ABL gene and the RAS gene, as well as the SRC gene that we already talked about, that were in all cases derived from a normal cellular gene and altered in ways that made these genes behave not in a normal fashion, but to change the behavior of a normal cell to a cancer cell. So, proceeding on from this moment of revelation that there were a lot of retroviruses that could cause cancer in animals that carried normal genes, it became apparent that it was important to look in cancer cells from humans to see whether any of these genes might be altered and indeed driving the cancerous behavior of human tumors. And by 1990 and increasingly thereafter, it became apparent that many of the genes that had been discovered by using retroviruses were actually important in the causation of human cancers because they underwent mutations that changed the functional properties of those genes. And by 2000, as I'll illustrate in a moment, some of those cancers could be treated with drugs that were targeted against the mutant proteins that were believed to be driving the human cancers. The most dramatic of these, in many ways, was work with the drug called IMATINIB, or commercially GLEEVEC. By 1999, it became apparent from clinical trials that this very small molecule, which is orally available, blocks the action of an oncogenic enzyme derived from a gene that I mentioned on a prior slide called ABL, a gene that was first discovered because it appeared in a mouse leukemia virus, and that this gene was altered in virtually every case of a disease called chronic myeloid leukemia. And indeed, this drug, which blocks the enzymatic activity of the altered ABL protein, is able to turn a disease which was virtually uniformly lethal within 5 years of diagnosis into a disease that was compatible with a normal lifespan. The drug is continually required, but it's virtually non-toxic. It's active orally, so it doesn't require visits to a physician. It was also soon proven that this drug inhibits other rather similar enzymes that are also protein tyrosine kinases, and that some of those genes also cause rare cancers. And if this drug is used to treat those diseases, like gastrointestinal stromal tumors and some rare leukemias, the drug is useful in those cases as well. As you might expect, because human cancer cells are, as I'll describe in more detail in just a moment, continuing to acquire mutations and undergoing a kind of evolution, some patients become resistant to drugs, but there are similar drugs that have been developed that are able to overcome the resistance that results from secondary mutations in the ABL oncogene. Now, this has led to a dramatic change in the way we think about the origins of cancer, about the genes that contribute to it, about the ways we might treat cancer and try to anticipate it. I don't have time to discuss all those things, but I want to give you some sense of how the early experiments with Rous sarcoma virus that led to the discovery of the cellular SRC gene has changed things. So we know that cancers are all different. There are differences in between types of cancer that might arise in the lung or the pancreas or the colon or other organs, and we know that even within a single type of cancer, such as lung cancer, there are many different genes that might undergo the mutations that drive cancers as observed in individual patients. And these these cancers can frequently be treated these days. By frequently, I don't mean 70%, I mean 10 or 15 or 20%, by using drugs like GLEEVEC. And here are a few examples. One I've shown you already, the chronic myeloid leukemia, certain breast cancers that have an abnormality of another gene that encodes a protein tyrosine kinase, certain lung cancers, especially lung adenocarcinoma, and melanomas, as shown at the bottom, that are frequently driven by mutations in a gene called the BRAF gene. These mutations are also useful for the development of a new kind of therapy that I won't have time to describe today and haven't really worked on in any significant way, and that is immunotherapies. Immunotherapies are strategies for harnessing the immune system to respond to what we believe are the abnormal proteins generated by the mutations I've been telling you about. Those mutations make abnormal versions of our normal proteins and allow the immune system, under the right circumstances, to respond to them. And there are many promising immunotherapeutic strategies that are now being used, exciting a great deal of interest. Not a subject for today, but one that you should be aware of, not only as a therapeutic promise, but also as a way to illustrate the importance of genetic mutations as a way of understanding cancer and a way of approaching treatment through the modulation of the immune system. I also want to point out another feature that is critical to understanding cancer in a modern sense, and that is that cancers are evolutionary systems. They are the product of mutations that are generated by errors in the normal replication process of cells and by by exposure to mutagenic agents. And there is in the growth of a cancer the same kind of selective pressure that exists in nature to generate the heterogeneity of animal and plant species that we see all around us. And that's illustrated by recent work that's been done in other labs showing that while a cancer at its primary site may have a small, relatively small selected group of mutations, that when you begin to look at the secondary growths of cancers at sites of metastasis, that additional mutations are found, suggesting that those additional mutations are contributing to a higher level of fitness that results in additional growth and spread of these cancers. And in that sense, cancers are like the evolutionary trees first envisioned for the origin of species by Charles Darwin 150 or more years ago. So let me just say a word about what I've told you about then being the latter part of the 20th century, where we learned that cancers are driven by mutant cellular genes, many of which were discovered as progenitors of the oncogenes found in retroviruses, and some of these have proven to be useful as explanations of human cancers and targets for new therapy, and that the mutations can be found in certain patterns that I'll say a little bit more about shortly, and that you actually can judge from the kinds of mutations that arise what kind of mutational influences may have given rise to those patterns of mutations. And all of this together has given rise to an idea that no longer do we diagnose cancer simply by looking under a microscope to see what kind of tissue the cancer has arisen from and what kinds of cellular properties might be evident under the light microscope, but we need to look under the lens of DNA sequencing and analysis of genomes and gene expression to learn about the molecular category that a cancer belongs to, because that's a guide to how therapy might be undertaken and what the patient's chances of survival might be. A whole new way of thinking about pathology that we often refer to as molecular pathology. So here's one example of how that might look. This is a colorful display of a couple of hundred lung adenocarcinomas, a very common form of cancer and a very common cause of cancer-related death in the human population everywhere in the world. And if you look at the results of a genetic analysis carried out by, in this case, at Harvard Medical School, you see that there are patterns of mutations that look extremely heterogeneous, but certain genes that we know from experience are commonly mutated in human cancers. Many cancers have mutations in those genes, like a gene called RAS that I've alluded to earlier, a gene called EGFR, which is also commonly mutated in cancers of the lung and the central nervous system and the colon, and quite a few others. Then there are other genes that are mutated at a very low frequency, and it's a little hard to know whether they are actually major drivers of cancer or simply along for the ride. This kind of analysis has given rise to a new way of thinking about cancer and indeed about other diseases as well, and has driven the notion that cancers are represent a kind of disease entity in which medicine can become much more precise. And in this new ecosystem, if you will, you can, at the center of it, we see a database, a knowledge network that is composed of information gathered from a variety of technologies, including genomics, but other disciplines as well, and that information gathered both by clinical observation and by work in the laboratory gives rise to a system in which clinical work suggests problems to be solved, laboratory work helps to solve them, and that ends up influencing the way in which we approach the diagnosis and treatment of disease. From my perspective as a working scientist, we often think that the kinds of ideas and experiments that we pursue are initiated with a study of the genomes of cancer, that those may influence the kinds of therapeutic strategies we might take, and that by making observations from both of these domains, we begin to think about how to explore the biology and genetics of human cancer. So I'd like to begin to wrap this up by telling you a little bit about current work in my own group that constitute three examples of now, how we work in this current era, understanding that cancers are generated by genetic change, that cancers are undergoing evolution, that cancers change the behavior of cells in fundamental ways, and that we're struggling to understand what these patterns of mutations mean, how cancer is generated, and how our understanding of how cancer arises might create new opportunities for trying to treat it. I'm going to say a few words in the last 10 or 15 minutes or so about how observations that were made by using cancer genomics, specifically in general from a project initiated at the National Cancer Institute over the last decade called the Cancer Genome Atlas, or TCGA, are influencing the new research directions that are being undertaken in my own lab, where, as I'm fond of saying, for almost 50 years we've been pursuing the same basic question of how a normal cell becomes a cancer cell, but we're pursuing that question now not by using a virus from chickens that makes an unusual form of cancer, but by using the modern tools of genomics and cell biology and other techniques to try to understand cancer by working directly, in most cases, with information we gather from human cancers, not chicken cancers. The first is to try to identify and understand the new cancer genes that are identified through genomics. The second is to try to understand the meaning of these complex constellations of mutations that are often seen, or the absence of such combinations. And thirdly, by defining conditions that allow us to understand more precisely why certain kinds of cancer, like colon cancer or brain cancer or lung cancer, tend to be associated with certain kinds of combinations of mutations. What does that mean, and how might that change our thinking about the prevention and treatment of cancer? So let me just say briefly a few words about something that is a fairly general concern, and that is, how do you interpret mutations that are found with genomics technologies that implicate genes we would not necessarily have suspected as being candidates to be cancer genes? Here's one example that my own lab has worked on for a couple of years, and that is mutations that have been found in a wide variety of cancers that affect the genes that make proteins that allow early RNA, so-called pre-RNA, to be spliced. You all know, I hope, that genes are composed of introns and exons. I mentioned that earlier, that RNA made from those genes has to be processed by a complex machine called the spliceosome to make transcripts that can be read by the ribosomes to produce proteins. We now know of many proteins involved in such machines. At least 3 or 4 of them are commonly mutated at characteristic positions in each gene to make mutations that are observed with significant frequency in a wide number of cancers. One such cancer is the myelodysplastic syndrome, in which the 4 genes illustrated here are very commonly mutated. What can that possibly mean? What kind of results might come from such an observation? Does it mean that there is some specific gene whose transcript is abnormally spliced so that it generates a cancer-causing protein? Or does it mean that there is some other function associated with these proteins that we don't understand? There are some simple questions to ask. How do such mutations affect splicing and how does it affect the profile of messenger RNA in a cell. A postdoctoral fellow of mine worked on that for some years, tried to build models for cancer that were dependent on these mutations with some success, but not sufficient success to try to answer the really critical questions. That is, what kind of property of cancer cells are conferred by such mutations? And are those mutations required to maintain the cancerous state of the cell? That's a critical element, because if you want to treat cancers, you have to know that you're addressing your therapy to something that's required for tumor maintenance, as we talked about in the context of SARC some time ago. And is there a therapeutic opportunity afforded by this? And these are questions that we have been unable to answer. And with the departure of Dennis Fay from my group, indeed, we have stopped working on this problem, but it does illustrate a fundamental question. When you see a gene mutated in a large number of cancers, presumably that class of mutations contribute to cancer in some way. What is the property that's being conferred, and is that a useful property to understand? The second example has to do with finding some kind of meaning in mutational combinations. We know that certain combinations of mutations are frequently found in certain cancers, that some combinations are almost never found despite the expectation that they would be found. Do these things have meaning, and can we exploit them? One example is found in that common cancer, lung adenocarcinoma, where we know that mutations affecting the gene called KRAS are found in about 30% of cancers, and that about 15% of tumors have mutations in the gene called the epidermal growth factor receptor gene. And yet, as shown by these bar graphs, while the mutations are themselves frequent, they never occur, or almost never occur, in the same tumor. Why is that? Well, you could imagine that if one gene is mutated, the other will not be mutated because they work in the same so-called signaling pathway. That was a popular explanation. Or it might be that a cell that has mutations in both of those genes has a disadvantage with respect to its fitness, and hence the combination is selected against. And one way to approach this, out of several that we tried, is to try to co-express those two mutations in the same cell. And when we do that in a human lung cancer cell line that has mutation in one or the other of the two genes, and we express the other gene, then we find that the viability, the fitness of those cells is impaired, suggesting that coexistence of the two mutations is a toxic situation in which cells are disadvantaged by having both of these genes expressed. Well, why might that be? Well, here I try to introduce you very quickly to the so-called signaling pathway in which the epidermal growth factor receptor and RAS both work. And we know from biochemical experiments, I don't have time to show you, that the kinase called ERK, which is the colored circle near the bottom of this pathway, is very quickly activated and may be a central component of this pathway. And that raises the question of whether inhibition of the protein that activates ERK, namely the MEK kinase, might be a target for interfering with this pathway. And indeed, if you use an inhibitor I see it's in the wrong place, it should be down below affecting MEK, that you in fact protect cells from toxicity that's produced by excess signaling through this pathway. And that's shown in the bar graphs on the right, a little difficult to see because of the coloring here, but the number of cells is restored to normal if you add an inhibitor of the MEK kinase at the same time that you induce a high level of signaling through RAS. So that begins to suggest that this pathway could be manipulated to try to kill cancer cells, for example, by recognizing that in addition to signaling pathways working through direct activation, the pathways are also regulated by so-called feedback mechanisms. And you might imagine that if you could block a feedback mechanism that normally restricts the activity of a kinase like the ERK kinase, that you could actually generate a toxicity in a cell that's expressing one of the an inappropriately activated component of the signaling pathway. So in the presence, for example, of a RAS mutation, blocking the activity of a feedback enzyme like the so-called dual activity phosphatase, DUSP, that you could make cells toxic. And indeed, in work I don't have time to show you directly, it is possible to show that cancer cells that make a mutant RAS protein become intoxicated by inhibition of feedback mechanisms that govern the activity of a protein like the ERK kinase. The third example I'd like to tell you about briefly is one that has to do with trying to understand the relationship between mutational profiles and different developmental lineages. And we've done that with a test case, small cell lung cancer, which accounts for about 15% of lung cancers worldwide. The cells are neuroendocrine cells. The cancers are highly lethal. Most patients, well over 90%, dying within the first year of diagnosis. No new treatments for over 30 years. And these cancers are distinguished, first of all, by having the markers of being neuroendocrine cells, and because most of the cancers have mutations of a class of cancer genes known as tumor suppressor genes, in particular the two very commonly affected tumor suppressor genes known as the RB or retinoblastoma gene and the P53 gene. But these cancers have been hard to study generically because they tend to present in the clinic at a time when the cancers are already advanced. It's hard to get fresh material to examine, and it's been a goal for some years to try to identify better materials in which to study the way in which these cancers are generated, how they're controlled, and how they might be treated. And we've taken an approach that is unusual but not unprecedented of trying to generate these cancers by starting with human embryonic stem cells, differentiating those cells to make the likely cell of origin, in this case a neuroendocrine cell, and then to make mutations that have or have not been associated with these cancers to see whether we can recreate the initiation of the cancers in culture. So to do this, we've used an embryonic stem cell line from Rockefeller University. There's a kind of symmetry here. Rockefeller University now providing a cell line as opposed to Rockefeller University 100 years ago providing Rous sarcoma virus. And we use tools worked out by others for making lung cells from these embryonic stem cells. And then we use the markers of differentiation to try to characterize the cell types that are generated and then use the cells that appear to be lung cells to try to recreate lung cancers by introducing the mutations that are associated with different kinds of lung cancers, adenocarcinoma, squamous cell carcinoma, or in this case, small cell carcinoma. And our focus has been, as I've mentioned, on small cell lung cancer, looking for cells with neuroendocrine markers like c-src, which will be the one we'll use in this discussion, and we look to see what happens when we deprive those cells of the tumor suppressor genes, RB and P53, that I mentioned a moment ago. The first important step in trying to do this was a fortuitous observation that by inhibiting a signaling pathway called the Notch pathway, by a method I don't really have time to describe, you can change the behavior of the cells in culture that have been differentiated from a stem cell to make a neuroendocrine cell that stains red because it expresses c-src, a marker of neuroendocrine cells. And this is simply a quantitative assessment of how the number of neuroendocrine cells, which is extremely low in cultures that are simply characteristic of lung cells, but not of neuroendocrine cells, from less than 1% to roughly 8 to 10%, and that's shown in a bar graph and also by cell sorting. If you reduce the expression of the tumor suppressor gene, RB, you can increase the percentage of the pulmonary neuroendocrine cells from about 10% to nearly 50%. That's dramatic. And then if you inhibit as well the tumor suppressor gene P53, as well as having interfered with signaling by the Notch pathway, having reduced expression of the RB gene, then the cells that were derived from the human embryonic stem cells will form tumors in mice, and that's shown in the numbers below. For some reason, the coloring is not turning up here. And these tumors, shown in the upper left, when characterized by various markers, are clearly lung cells. They're not teratomas. They have the markers such as CGRP that are characteristic of neuroendocrine cells. These are not full-fledged small cell lung cancers. They're not yet invasive, so we need to understand how progression occurs, but they are representative of an early stage small cell lung cancer that allow us to begin to probe how such cancers arise. So one final comment. Let me emphasize a transition here that may be apparent to some of you, that 50 years ago or so, when people like me began working on cancer, we had to approach our efforts to understand cancer by working with chickens, not humans, a cell type that rarely accounts for human cancers, fibroblasts, and used viruses of chickens, a kind of artificial way to study cancer, to look at transformation in culture. But today we can start with human cells from an embryo, differentiate them using available tools, make the presumptive precursor cell for such cancers, and then turn them into early stage cancers, such as small cell lung cancer, by using tools that recapitulate the kinds of mutations we find in human cancers. So that is a significant advance in thinking about how we approach cancer as scientists using experimental tools. People who do this kind of work are critical. Here is Dennis Fay, who did the work that I mentioned briefly on splicing factor mutations. Arun Uni and Will Lockwood, who've done the work on the toxicity that's generated by inappropriate levels of expression of the RAS signaling pathway, and Joyce Chen, who's done the work on initiating small cell lung cancer. For all of us, you recall that when I spoke on Friday, we talked about the internationalization of science. Science is inherently a team sport that is played throughout the world, and that sport, in my way of thinking, is a effort made by the scientific community to combat a disease that afflicts everyone around the world, and by combining the right attitude towards scientific work with the new technologies that have become available over the last few decades, I think that cancer is increasingly coming under control and will be even more so in the future. Thank you again very much for your attention and for this visit to Lebanon. Be happy to take some questions if it's not too late. I know some of you have classes at 11 o'clock. I'm not sure what time it is. That's not 11. That's not right. Yeah, it's not 11:30. It's 10:30. Down here.
Thank you very much for this extremely interesting lecture. So my question is, it's a bit of...
Why don't you wait for a microphone? I can hear you, but I suspect people in the back have no idea what you're saying.
So thank you very much for this extremely interesting lecture again, Dr. Varmus. So 2 years ago, I think it was a couple of years ago, work came out of Sloan Kettering that showed for the first time that in the case of breast cancer, scientists were able to detect metastasis very early on, so as opposed to what we used to think historically. So even at the level of few cells, when it comes to breast cancer, it actually was able to undergo metastasis. So how do you, what do you think, how should we rethink, you know, our view or reshape how we think about metastasis, or how would that redefine the idea of benign versus malignant, which we've been following for
Well, this
years?
is a very good question. And actually, evidence for very early stage metastasis is much older than just 2 years ago. Indeed, when I was at Sloan Kettering in the late 2000s, we published some work on early metastasis in which cells were migrating and taking up residence in the lung after the induction of cancers in the mouse breast, and would remain latent for long periods of time before they would produce large growths. And we've seen there's a great deal of evidence for phenomena like that in human patients. Interestingly, that kind of pattern is particularly characteristic of certain cases of breast cancer, much less common in some other cancers. So there may be constellations of mutations and inherited genetic properties that contribute to the tendency for breast cancers, in some cases, to escape from their primary site, go to distant sites, but not have the potential to grow in those distant sites until something else happens, perhaps an additional mutation or some change in the environment that induces genes that recruit blood vessels or allow cells to become invasive or release cells from growth constraints. So there are a set of questions. Why do cells migrate to some sites and not others? What has to be present at the site to which a cancer cell migrates that allow the cells to settle there and establish residence and then continue to grow later on? And why do some cells keep growing when they migrate, and why do some cells wait until additional events have occurred? Many people have ideas about how this might work. You're correct, and I appreciate your drawing attention to the fact that understanding metastasis is a critical element in trying to control solid tumors, because it's not the primary tumor that generally results in death. There are situations in which it does, but more commonly, cancers kill because they metastasize, and even if the primary tumor is removed, patients die because of growth of cancers at metastatic sites. I'm sorry to say that we don't understand what it is that accounts for either the appearance of cancer cells at certain sites, what produces latency, and what accounts for the latency in many cases, and what accounts for regrowth. If we could probe those more compellingly, as I think we now can with some of the new tools that are available, especially cells that are derived directly from patients, so-called patient-derived xenografts that can be put into mice and observed for their metastatic potential, and using the tools of genomics and proteomics and other high-throughput methods, perhaps we'll make some progress against the many unsolved problems. I've been telling a story. It has some outcomes. The outcome we all desire, that all cancers be controllable, has not yet been achieved.
Thank you very much, Dr. Varmus, Gadel Hashfulihan, Faculty of Medicine. As an endocrinologist and thinking humoral
Can you
spread.
hold the microphone a little bit away from you?
Okay, as an endocrinologist and thinking humoral spread, can you comment on the relationship between the microbiome and all the survival cancer genes effect interaction?
No. I'm not a practicing oncologist, but I can comment
on that. Clearly, the influence of the microbes that inhabit our body, especially in our gut, but other places as well, can have an influence on cells elsewhere in the body. There is a lot of interest in pursuing that idea because of some provocative early experiments, but I think we don't understand it well enough to make any clear predictions at this point about how the microbiome affects cancer cell growth or metastasis, or in particular, what we should do about it. I would not recommend at this point changing your microbiome because you've been diagnosed with cancer, but I do recommend that people who are expert at analyzing the microbiome and the products that it releases into the bloodstream pursue the possible connections between the microbiome and cancer growth. There's a question here.
Yes.
Got a microphone?
I'm Nadine Darwish. I would like to welcome you to Lebanon. I would like to emphasize that many faculty members at AUB were students and postdoctoral researchers when you were at NIH. I was in the lab of Dr. Stuart Yespa.
Who?
Stuart Yespa,
yes,
Stu.
great guy.
So my question is more rather political question, science and politics. We know what's going on with the research, environmental-driven research these days. What about biomedical research? I mean, we don't hear as much as we do about biomedical research versus environmental research in the current administration.
Okay, well, that obviously a question more appropriate for last Friday than for today, but I'm happy to answer it. The question is, what's happening to biomedical research in the era of Donald Trump? I think that's your question. And the answer is actually is fairly encouraging, that while Donald Trump's administration attempted to reduce the NIH budget by several $billion when he first arrived, I think they've learned that there is tremendous public and congressional support for the NIH. And efforts to reduce the budget have backfired, that Congress has stepped up and actually increased the budget over the last few years. The prior director of NIH has stayed on, so his job is not subject to being filled by an incompetent person. The president has another appointment to make. That's the director of the National Cancer Institute, filling the job that I previously held. And by a mechanism that most of us don't understand, someone extremely qualified has been appointed to that position, someone named Ned Sharpless. So the NIH is being run by good people. It's being supported financially by the NIH. It still has strong public support. Almost miraculously in the recent shutdown was protected because the appropriation bill, which of course comes from Congress, that supports the NIH and the rest of the Department of Health and Human Services, was passed earlier in the year. So the Department of Health and Human Services and the NIH were not affected by the shutdown, not affected directly. Indirectly, of course, because many other science agencies were shut down for a few weeks. Among those, the National Science Foundation and other parts of the government with which NIH scientists collaborate. But also, the shutdown does have a negative connotation about the commitment of the administration to keep our government operating. Government is essential for the conduct of science in the U.S., especially basic science, and no one is happy about the vulnerability that we face this year and next year in the future as a result of conflicts over building a wall between Mexico and the U.S. that should not be the reason for shutting down the scientific establishment in the U.S. for prolonged periods.
Thank you for a wonderful lecture. Well, with all the advances at the genomic level and identifying new genes and new oncogenes to be targeted in different patients within every tumor type, and the advent of the era of personalized medicine, the problem remains actually developing the drugs to target those. And with pharmaceutical companies following the model of trying to target the widest range of patients possible, how do you think that shift in modus operandi and in the paradigm of drug development should happen in order to meet that need?
Yeah, well, you're drawing attention to one of the things that is motivating the drug industry right now, and I think appropriately modifying it. My own view is that this has been a healthy thing for drug development because it's a rational approach to obtaining drugs. You know, for many years, the way in which you sought cancer drugs was to take as many compounds as possible and to screen them against a multiplicity of cell lines. Now it's possible to work with a mechanism-based approach and have a higher level of success. And indeed, we've had many successes with targeted therapies. While I've tried to be careful to point out that only a minority of patients are benefiting from those therapies, and indeed, many patients who benefit only for a short period of time until the evolution of the tumor produces a cell that resists the effects of the drug. So these drugs are not cures, they're not panaceas, they're very far from perfect. My major concern is that there's so many genes that have been implicated in cancer. Frequently we don't know, as I tried to emphasize in my discussion about splicing factor mutations, we don't actually know what they do, how they work, and how to design the therapies. Moreover, when a drug company pursues a target like a splicing factor mutation, it does so at considerable risk because we don't know how effective these drugs will be, what kinds of side effects they'll have, how long they'll last, and sometimes you end up with drugs that are effective for the treatment of cancers that affect only a few hundred or a few thousand patients per year, in which case the financial model becomes difficult. I think we all take encouragement from what happened with GLEEVEC, the drug that I described in a little bit of detail. Initially, and I know I was there running the National Institute of Health at that time, the company was concerned because there are only 6,000 cases of chronic myeloid leukemia in the US each year, 15 times that many roughly in the world, and that's not a lot of patients, but these drugs work by controlling cancer, not curing it. That means that all patients take it for a lifetime. Secondly, as I mentioned, this is a drug which is active against a variety of protein tyrosine kinases and can be used to treat other cancers as well as chronic myeloid leukemia. And indeed, GLEEVEC has become a multi-billion-dollar drug. So while the drug remains expensive in most parts of the world, at least it will be expensive until it comes off patent, which will happen almost immediately. It sets a precedent that encourages investment in companies that are making targeted drugs. So I think on the whole, it's been beneficial to watch the success of GLEEVEC.
Yeah, thank you very much for this inspirational talk. I want to come back to the question of the evolution of the cancer genome.
Sure.
And those mutations that are being fixed. From an evolutionary perspective, what would be the advantage or fitness that is brought other than the proliferative effect at the cell level?
It's getting muddled back here.
Yeah, what would be the advantage that is brought by having those mutations fixed with time, other than the proliferative effects that we see on the cells?
There's no advantage to the host. The host is going to die as the cancer evolves. But the selection here is not a selection that affects the descendants. What the evolutionary process does is represent the advantage to the cancer cell to be dominant. So the cancer wants to grow, and if it grows and outgrows its neighbor cells, it will become a collection of cells that ultimately does itself no good because it kills the host in which it's growing. But the evolution is driven by the dominance of the cell in which these mutations have occurred that allowed that cell to become the majority player in a tumor that is growing at one site and spreading to other sites. So evolution doesn't really have a purpose. Evolution is driven by forces, and in this case, there are more cells if cells grow better. And if they die less frequently, more likely to trigger a cell division cycle, are able to spread to other sites. So as in the case of species evolution, it's survival, fitness that dominates the outcome of the evolutionary process, not the benefit of the host.
Thank you very much, Professor Varmus, for a very inspiring lecture, and thank you all for a very active participation. Thank you. and welcome to AUBM. Can we have a picture with the audience? Please remain seated.